All right, so welcome back to the next part in our cell lecture. So in this part, this is part 6, we're going to be looking at something called molecular transport. So molecular transport in general is going to be this process of moving molecules and moving ions, and then we'll also talk about moving water, but moving all of these things into or out of the cell. And we'll talk about what influences the movement, and why things move in particular directions. Okay, so the first thing to talk about is this particular type of motion of ions and molecules called Brownian motion. Okay, and Brownian motion essentially states that molecules are going to move at random as a result of random collisions with each other within their given space. So within their environment, molecules are going to move randomly, and because they move randomly, they randomly hit other ions, and because they hit other ions and other molecules, they're then going to move again. Okay, and so this random movement is called Brownian motion. All right. Now, when you have a group of molecules that are highly concentrated in one area, there's going to be a tendency for them to move via this Brownian motion, move down their concentration gradient, right? So move from where they are highly concentrated to where they are not as highly concentrated. Okay, so here you can see on the left, we have the left beaker. We have a group of ions or molecules that are highly concentrated in one area of the beaker within the water, right? Well, following the principles of Brownian motion, we're going to distribute those ions. We're going to distribute them out from their highly concentrated area to a more evenly distributed area. Okay, and we're going to call this process passive transport. Okay, when you see the term passive from now on, think that this does not require energy. Okay, there's no ATP requirement in order for this to happen. Okay, you just move down the concentration gradient. A great example of this is going down a slide, right? So think back to when you were in elementary school and you go outside on a hot day and you slide down those metal slides that always, burns your legs as you go down. When you go down the slide, you don't have to put in any energy, right? You have gravity doing the work, right? You're using your body weight and you're sliding down the slide, okay? Likewise, in this process of passive transport, we're moving down a concentration gradient, okay? That's what we're calling it. So we move down the concentration gradient from where we're highly concentrated to where we are not as highly concentrated, okay? And so we're going to talk about something called osmosis, and we're going to talk about something called diffusion, okay? So this is passive transport, right? So this is the passive movement of ions. If you have it require energy, if you have this process require energy, now we're going to call it active transport, right? So imagine you're back on the playground when you're young and you're going down a slide, right? When you go down the slide, it's very easy. It's energy free. It's not that hard. When you were always on the playground and you would try to go up the slide, right? You'd try to start at the bottom, you tried to climb up, right? And your teacher would yell at you. So when you try to climb up the slide, now that's requiring energy, right? So this is called active transport. You have to invest energy into the system, and now we're moving what's called against our concentration gradient. Okay, so now we're moving from where we are from a lower concentration to a higher concentration. So we're trying to go to where there's already more of a particular molecule or something like that. Okay, so in this case, we're going against the concentration gradient. We're moving up it. All right, and this is going to be more difficult to accomplish. Okay, because it's going to require the use of energy. All right, so when we look at active transport, we can have different types of active transport. Okay, so there's something called primary active transport, and there's something called secondary active transport. Right. So primary active transport is the direct use of ATP to move molecules or ions against their concentration gradient. Okay, so we're directly using ATP. If we're using secondary active transport, we're not using ATP directly, but we're using another ion's concentration gradient to help move another ion against its concentration gradient. Okay, so in other words, the concentration gradient made by primary active transport is used in secondary active transport. okay? And that doesn't make a whole lot of sense right now, but it will make more sense later whenever we look at some examples, okay? So the idea here is active transport, we're using energy. Passive transport, we are not using energy, okay? All right, so again, depending on how a molecule or an ion or something is charged and what its solubility is and whatnot, and whether it's permeable or impermeable to the membrane, this is going to influence how they move, okay? So does it move with the use of energy? Does it not? Does it use a transport protein? Or does it just diffuse directly through the membrane? Okay, so review this table. This is a similar table to what we looked at in a previous part of this lecture. Okay, so the first type of movement, and we're going to start off talking about passive transport, is called simple diffusion. Okay, so diffusion, when we're talking about diffusion specifically, we're talking about a solute. Okay, so a solute is an actual thing, it's an actual molecule of something. So a molecule of sugar or a molecule of sodium, so an ion, something like that, as opposed to a solvent. So a solvent is the liquid, okay? The solvent is what the solute is dissolved in, all right? So when we're talking about diffusion, we're talking specifically about the solute. So in simple diffusion, we have the movement of the solute down its concentration gradient without the need for a transport protein, okay? Without the need for a transport protein. We're going from high to low. They can move independently of each other, and they are able to move without the use of any type of help. Okay, no transport protein is needed. All right. So if we look at this top row, we can see that this dotted kind of blue line, this is representing our selectively permeable or our semi-permeable membrane. Right. So remember, I like the term selectively permeable a little bit more. So in this case, we're highly concentrated on the left side of the membrane, and we have none on the other side. So by principles of Brownian motion, we're going to move and diffuse down our concentration gradient until we have, let me go back, until we have equalized their concentrations, okay? So that's the goal here, okay? Now, it doesn't really have a goal in mind, like it wants to do this, but this is what we're going for, right? We're trying to equalize the concentration of solute on one side of the membrane versus the other side of the membrane, okay? You can see the same thing at the bottom, but now we have two. So we have high concentration of red ions on this side, high concentration of blue ions on this side, and they're going to move until they equalize across this membrane, okay? Now, one important thing to keep in mind is that once they equalize, so in this case up here, they're equalized, in this case down here, they're equalized, they still are moving, okay? But they're moving with a net zero flux. Okay, so this concept here, zero net flux or net zero flux. What this means is that the net result is essentially nothing, okay? But you still have some movement, okay? So you still have some things moving from left to right, you still have some things moving from right to left, but the overall effect is that it's equalized and we have moved down our concentration gradient, okay? So this is simple diffusion. We're going freely across a selectively permeable membrane without the use of any transport protein or anything like that, okay? So here's a good example of iodine and water, right? So this bottom picture here, this animation, this is iodine and water, right? So you drop the iodine in, and you can see immediately as the iodine hits the water, it's highly concentrated in the area where it first enters. And then over time, that iodine begins to spread out via Brownian motion into the glass of water, and it is evenly distributed, right? So because of those random collisions and random movements, you get this nice distribution of solutes, okay? All right, the second type of transport is called osmosis, okay? So diffusion, we said, diffusion was dealing with the solute. So it was dealing with the actual thing, the molecule, all right? With osmosis, we're not talking about the solute now we're talking about the solvent. So in this case, we're talking about water, okay? So the membrane, this selectively permeable or semi-permeable membrane, is not permeable to the solute, okay? So it's impermeable to these ions or these molecules. So what that means is in order to equalize their concentration, we're not going to be able to move the ion or the molecule, so instead we have to move the amount of water, Okay, so we have to move the solvent. All right. And so what happens is water is going to move toward the area that has a higher concentration. Okay, so if we look at this beaker on the left, which side of the membrane, left or right, would you say is more highly concentrated, right? Well, hopefully you would say the right side, right? We have the same amount of water on both sides, but there are far more molecules on the right side than the left. So this right side is more concentrated than the left. So what this means is water is going to move from the left to the right in an attempt to equalize out and to equalize the relative concentration. Okay, so we're not changing the number of solutes on the side because we said that the membrane is impermeable to the solute. So there's no way you can change that. But what we are going to change is the amount of water on one side versus the other. And thus, when we change the water, we change the concentration. Okay, so you can see here in the image to the right now, water has moved from the left over to the right, and it has now caused a equal concentration. So proportionally, given the amount of water and solutes, both of these sides here are equal. Okay. Now, this water can move depending on the osmolarity of cells. So we'll talk about something called isotonic, hypertonic, and hypotonic solutions, and this will influence the movement of water with respect to the cell. Okay, so here you can see, depending on which side is more concentrated, we're going to have a movement of water from one side to the other. So in this illustration, we're moving from the left side of the beaker over to the right, and you see it equalize concentration. Okay. All right. So talking about this idea of osmosis and this the osmolarity or the tonicity. Okay. So there's three different types here. There's isotonic, hypertonic, and hypotonic. Okay. Now, what we're talking about here is the concentration of solutes inside the cell relative to the concentration of solutes outside the cell. Okay. And again, following the same principle of what we just talked about, water is going to move to where there is a greater or higher concentration of solutes. Okay. So in this case, in an isotonic solution, iso means the same, or they're equal. So there's equal concentration inside the cell versus outside the cell. Okay. Now that doesn't necessarily mean that the specific number, the actual number of solutes on the inside versus the outside is the same, but relative to the amount of water and proportional to the amount of water, they're equal. Okay. So this is what's used in clinical settings as normal saline, right? If you just want to replace fluid volume, okay, you can give 0.9% sodium chloride, normal saline. This is not going to cause any massive movements of fluid into or out of cells, okay? And that's a good thing in this case, okay? No net movement of water. The key word there is no net movement, okay? You still have movement of water. Water's still going to move in. Water's still going to move out, like you see in this little illustration down here, but it's going to be relatively equal. So the net is nothing. A little bit goes out and a little bit comes back in, okay? All right, the second type of solution is a hypotonic solution, okay? In this case, we're referring to the inside of the cell being less concentrated than the outside, okay? So there's a higher concentration. I'm sorry, I said that backwards. There's a higher concentration on the inside of the cell than outside. So what that means is water is going to rush into the cell because on the inside, there's a very high concentration of solutes. We want to equalize that concentration on the inside relative to the outside, so water is going to come in. What this is going to cause the cell to do is as it collects and takes on more water, the cell is going to get larger, it's going to swell, and if it occurs to a great enough extent, it can actually cause the cell to burst, okay? When a cell bursts, that is called lysis, okay? Lysis is the bursting or the rupturing of a cell. If this happens specifically to red blood cells, we call this hemolysis, okay? Hemo or heme is referring to blood, and lysis is referring to the bursting or rupturing of, so this is the, hemolysis is the rupturing of red blood cells, okay? This is what would happen if you put red blood cells in pure water, okay? You put a red blood cell in water, there's relatively few, if any, solutes in the water. but there are lots of solutes in the red blood cell, so the water in the environment is going to rush into the cell very rapidly and cause that red blood cell to burst and to lyse. Okay. All right, and then the last one is hypertonic. Okay, so if you know what hypotonic is, then hypertonic is exactly the opposite. In this case, there is a higher concentration of solutes outside the cell relative to the inside. So water's still going to move to where there's a higher concentration, but in this case, it's going to move out of the cell because there's a higher concentration outside, leaving behind less water on the inside of the cell. And if this happens to a great enough extent, it can cause the cell to shrivel and shrink. And this is what's called crenation. So the cell is said to have crenated or shrinking and shriveled up. Okay, this is what would happen if you placed the red blood cells in a 3% saline or 3% sodium, 3% sodium chloride solution. The solution of that solution has a greater concentration of solutes than the red blood cell, and so water naturally is going to move out of the red blood cell and into the surrounding environment, causing the cell to shrink and shrivel. Okay. All right. So you can see this example here. Now, one important thing for all of these, you're going to have movement of water in and out, but it's going to be greatest in one direction or another for hypertonic and hypotonic. And then for isotonic, you have a little bit come in and you have a little bit go out. So they're going to be more or less equal. If you look at these images down here, you can see that in the hypertonic solution, all of these cells are very shriveled. They kind of have these pointed edges. In the isotonic, you see normal red blood cells with a kind of depression in the middle. And then on the right, you see red blood cells that are extremely large in size because they have swollen and they could potentially get ready to burst. All right. Okay, so we talked about simple diffusion. We talked about the movement of water, right? Those are both types of passive transport. The third type of passive transport is facilitated diffusion, okay? Facilitated diffusion utilizes no energy, just like simple diffusion. But in this case, instead of the ions freely going back and forth across the plasma membrane, now we have to use some type of transport protein, okay? So this can be a channel protein, or this could be a carrier protein, all right? So you have a channel protein or a protein channel that occurs when you have a protein open up, and on the inside of this protein, there's a nice channel, and it allows a particular ion or molecule to flow nicely from one side to another. And so we'll see a lot of these in the form of sodium channels, in the form of potassium channels, in the form of calcium channels, as we move on in the semester, okay? And then carrier proteins are going to be taking our ions or molecules, binding them, and then moving them to the other side, okay? So it's still a passive process, but you're having to utilize a protein to do it instead of just having things move freely down their concentration gradient, okay? All right. So when we look at our protein channels here, we can see that we have different types, okay? And all of these are going to be examples of types of passive transport, okay? So the first one is called voltage gated. And now these are named according to their stimulus to be opened, right? So voltage gated is gated and opens in response to a change in voltage, right? Ligand gated, ligand is just a molecule that binds to the receptor, and then so it functions as both a receptor and a channel. So this is going to open in response to voltage. This is going to open in response to something binding to it, both B and C. Those are both ligand gated. And then D, this is a mechanosensitive. So this one opens and closes in response to physical pressure and physical manipulation of the channel. Okay, but all of these, when properly stimulated, will open up and allow ions and allow molecules to flow from one side to another. Okay, and we'll look at various examples of this throughout the semester. Okay. Okay, so as I was saying, all of those examples and all of those instances that we just talked about there were examples of passive transport. Now, these next few slides to finish this part of the lecture are going to talk about active transport. So if passive transport is moving things down their concentration gradient from high to low, either with a protein uh, uh, transport a protein, or without, you're still using no energy. Now, when we talk about active transport, we are using energy in some way. Okay, so here we're using a sodium potassium pump, and this is a great example of primary active transport. Okay, so what we're doing is we're taking sodium on the inside of the cell where it is not as concentrated, so it's highly concentrated on the outside of the cell, not as concentrated on the inside. And we're taking potassium ions from the outside of the cell, and we're bringing them back into the cell where they're highly concentrated on the inside. Okay, so if we look here at this first part, we're taking three sodium sodium ions, and we're moving them to the outside. Okay, they're highly concentrated on the outside, so by nature we have to use energy. We have to use ATP. Right. Likewise, we're taking two potassium ions, and we're moving them to the inside of the cell. Right. Potassium is highly concentrated on the inside of the cell, so we have to use energy. Okay, so moving these ions against their concentration gradient, we have to utilize ATP. Okay, we'll talk about this use of a sodium potassium pump. Sometimes it's called a sodium potassium ATPase because it's an enzyme and it uses ATP. We'll talk about this more when we get to our action potential lecture later in the semester. Okay. All right. So here you can see a proton pump, which is also utilizing ATP. This is shuttling hydrogen ions down, against their concentration gradient. And as it's doing that, it is having to utilize ATP here. Okay, so this is another example of a mode of active transport. Okay, utilizing energy. Okay, now here you can see what are called symporters and antiporters. Okay, so these are utilizing or one of these here on this example here, this is the sodium potassium pump that we just talked about. This is what's called an antiporter because it's moving things in opposite directions. And then here, this is a symporter. It's moving two things in the same direction, right? So this is one mechanism by which we can get glucose into the cell, right? So we use this sodium potassium pump that we just talked about, take sodium out of the cell, bring potassium into the cell. Now we have sodium outside where it's highly concentrated. Well, when sodium moves down its concentration gradient, that's just passive transport, right? And so sodium moving down its concentration gradient on the toward the inside of the cell can facilitate the intake of glucose into the cell. Okay, so this is what's called a sodium glucose symporter. All right, because we're utilizing sodium to simultaneously help bring in glucose into the cell. All right. So this one here is utilizing ATP indirectly because it's relying on this protein pump over here, which utilizes ATP directly. Okay. All right. So we're going to stop with this part of the lecture, and then we'll pick up with our last part by talking about some cell signaling, some different types of cell junctions, and we'll talk briefly about the cell cycle.